Usually when we talk about getting a medical diagnosis, there is this, I guess, this underlying expectation of precision, like it's engineering or something.
Right, yeah, like fixing a car.
Exactly. You fall, you break your arm, the doctor puts you in an x-ray machine, and there's that, jagged white line on the film. They just point to it and say, there it is.
Yeah, it's binary, broken or not broken. It's clean, and frankly, I think that's deeply comforting for us. We humans really, really like things to be visible and easily categorized.
Oh, totally. But then you step into the world of liver cancer, specifically liver hepatocellular carcinoma, or LIHC, and suddenly that metaphorical x-ray machine is it's completely broken.
It really is.
We're looking at a diagnostic landscape that is just incredibly murky. I mean, this is a highly aggressive malignancy. It is globally prevalent, and yet it's notoriously difficult to diagnose early enough to make a massive difference, right?
Yeah, that's the tragedy of it.
For years, doctors have been relying on blood tests, looking for serum biomarkers like alpha-fetoprotein or AFP. But AFP is, well, it's missing the mark, isn't it?
It is. I mean, AFP has been the standard for a long time, but it just lacks the necessary sensitivity and specificity.
Right.
In simple terms, it just doesn't always catch the cancer early, and sometimes it flags a problem when there actually isn't one. It is literally the definition of diagnostic muddy waters.
Which leads to those tragic missed opportunities.
Exactly. Missed windows for early intervention.
Well, clearing up those muddy waters is the entire mission of our deep dive today. We are exploring this completely paradigm-shifting study published in partnership with the Hormel Institute at the University of Minnesota.
And it's a fascinating one.
It really is. The researchers basically decided to stop looking the usual suspects. Instead, they cast this massive net into global genetic data and pulled out a completely hidden culprit in liver cancer progression. It's an enzyme called Squalene epoxidase, or SQLE.
Right.
So we are going to unpack this research step by step. We want to understand how a totally standard, cholesterol-building enzyme essentially turns liver cells rogue.
Yeah, and to really grasp why this is so groundbreaking, we need to set the stage by looking at what SQLE actually does in your body when you're healthy.
Okay, lay it on us.
So in the complex realm of cellular metabolism, Squalene epoxidase is what we call a rate-limiting enzyme in the cholesterol biosynthesis pathway.
Wait, let me stop you right there. A rate-limiting enzyme. That means it acts like a bottleneck, right? Like it controls the speed of the whole operation.
Precisely. If SQLE slows down, cholesterol production slows down. If it speeds up, production spikes. Its day job is simply to help your cells manufacture cholesterol. And to be clear, your cells need cholesterol.
Right. It gets a bad rap, but we need it.
Oh, absolutely. It's a vital structural component for healthy cell membranes.
Okay, so it's a completely normal, necessary biological factory worker.
Exactly. But what this study reveals is that in liver cancer, SQLE is not just doing its 9-to-5 day job. The researchers found a massive overexpression of this enzyme in liver tumors.
Wow.
And it bridges this gap that scientists have been trying to understand for years. I mean, the link between basic cellular metabolism and aggressive tumor growth. SQLE isn't just a bystander sitting in a cancerous liver, it is actively driving the malignancy.
Okay, let's unpack this, because anytime a study claims to find the hidden driver of a major cancer, I always want to know how they actually found it.
Right, the methodology.
Yeah, like they started with computer-based bioinformatics, right? What they call the in silico phase. But how do you even begin searching for one rogue enzyme in the entire human genome?
Well, you have to look everywhere at once, and you have to look for patterns that repeat. They didn't just study a small local group of 50 patients. They cast a massive multi-cohort dragnet. They pulled data from 9 independent data sets located in the Gene Expression Omnibus database, the GEO, and they combined that with the massive clinical data set from the Cancer Genome Atlas, the TCGA.
So we're talking about the genetic profiles of thousands of different people.
Thousands. And to sift through mountains of genetic code, you obviously can't use human eyes. They utilized highly sensitive mathematical algorithms specifically. One's called Lemma and DSEQ2.
Lemma and DC2.
Okay. Yeah, you can think of these algorithms as incredibly fine mathematical sieves. You pour thousands of genetic profiles through them, and they filter out all the normal, everyday biological noise.
Right, the background chatter.
Exactly. What gets caught in the sieve are the differentially expressed genes, the genes that are behaving wildly abnormally compared to healthy tissue.
So they pour the data from the first database through the sieve, And it flags what, like a few thousand suspicious genes?
Yes, initially thousands of genes looked a little strange in one dataset or another, but the power of this study is in the cross-referencing.
Oh, I see.
When they layered the results of all 9 independent databases over each other, looking for the overlap, SQLE was the absolutely only gene that was consistently upregulated across all 9 sets. Just one.
Wait, seriously? Out of thousands of genetic anomalies across 9 completely separate groups of people from different backgrounds, SQLE was the only one that was always hyperactive.
The only one.
That is wild.
It is. In the world of bioinformatics, finding a single gene that behaves identically across 9 distinct cohorts is basically striking gold. It completely overcomes the statistical biases you usually see in single cohort studies.
Makes sense.
And when they validated this against the Cancer Genome Atlas data, it confirmed exactly the same thing. SQLE is hugely amplified in tumor tissues compared to normal liver tissue.
But, just being present doesn't make it a good diagnostic tool. I mean, the AFP blood test we mentioned earlier is present, but it's not reliable. How do we know measuring this SQLE enzyme is actually a better way to test for cancer?
Because of a metric called the area under the curve, or AUC. This is how statisticians grade a diagnostic test's ability to distinguish between someone who is sick and someone who is healthy.
Okay, how does the scoring work?
A score of 1.0 means the test is 100% perfect, never making a mistake. A score of 0.5 means it's basically a coin flip. In this massive data set, SQLE scored an AUC of 0.884.
Oh, wow. So it is highly accurate at drawing a line in the sand between normal liver tissue and a tumor.
Incredibly accurate.
But let's look at the patients themselves, because if I'm looking at this data, I'm wondering who is most at risk. Does this gene expression change based on the patient's demographics? Like, is a 60-year-old man more likely to have a liver flooded with SQLE than a 40-year-old woman?
That is exactly what the researchers asked, and the answer is no.
Really?
Yeah, SQLE expression showed absolutely zero statistically significant relationship with patient age or gender.
So what triggers it to spike?
It doesn't correlate with who the patient is. It correlates entirely with how severe the disease is.
Ah, got it.
The researchers found that high SQLE expression was a massive red flag. It spiked heavily in patients whose tumors were poorly differentiated, meaning the cells had mutated so much they barely looked like liver cells anymore, what doctors call histologic grades G3 and G4.
Okay.
It spiked in patients who had an AFP over 400 nanograms per milliliter, and in patients who had advanced to pathological stage 3 and stage 4.
So it's basically a mirror reflecting how aggressive the cancer has become.
Precisely. And the survival statistics are grim but clear. High SQLE levels independently predict terrible outcomes.
In what way?
Well, patients with high SQLE didn't live as long. Overall, that's poor overall survival, or OS. The specific cancer was more lethal, meaning poor disease-specific survival, or DSS.
Right.
And the time before the tumor came roaring back after treatment, what doctors call the progression-free interval, or PFI, that was drastically shortened.
Okay, let's just pause and map this out to make sure we're holding on to the big picture here, because the sheer volume of data is intense.
It is a lot to take in.
So here's our first little summary. I'm picturing this SQLE enzyme as a rogue factory manager. This manager doesn't care if the factory is in a man or a woman, young or old. The manager's only goal is to aggressively expand the factory, the tumor.
I like that analogy.
And the data shows 3 main things so far. Number 1, they found this manager hiding using massive mathematical sieves like Lemma and DAC2 across 9 global datasets. Number 2, its presence is a highly accurate diagnostic marker with an AUC score of almost 0.9.
Right.
And number 3, when this manager takes over, it ignores all safety protocols, driving up the tumor stage and tanking the patient's survival chance.
Exactly. Spot on.
So if you are listening to this right now, think about this. If you were a doctor seeing this data, how would this change how you look at a patient's genetic screening? You aren't just looking to see if cancer cells are present anymore, right? You're looking for this specific metabolic engine that tells you exactly how aggressive that tumor is about to be.
Which brings us to a critical threshold in the scientific method. Data on a computer screen is compelling, but a computer model is not a human body.
Right, and I have to play devil's advocate here. I mean, correlation is not causation.
Always a fair point.
Just because we see this rogue factory manager standing at the scene of the crime, how do we know they're the one actually laying the bricks? Maybe the cancer's growing for some other reason, and the SQLE enzyme just happens to be accumulating as a byproduct.
And that is the exact skepticism researchers must have. They had to prove causality. So they moved from the computer into physical laboratories, what we call in situ and in vitro analysis.
Okay, getting their hands dirty.
Exactly. They started by taking actual physical human tissue samples from 48 patients. They used a technique called multiplex immunohistochemistry, or MIHC.
Oh, I love how visual this technique is.
Yeah.
They basically use chemical dyes to make specific proteins glow under a microscope, right?
Exactly. It's essentially color-coding the microscopic world. They used a red fluorescent dye to tag the SQLE enzyme. Then they used a green dye to tag a protein called pancytokeratin, or pan-CK.
Pancytokeratin, what does it do?
Well, pan-CK is a strict, universally recognized marker for epithelial cells. Those are the structural cells that line your organs, and crucially, they are the specific cells that mutate to form carcinomas.
So green means this is the structural organ cell, and red means this is the SQLE enzyme. What happened when they looked at the tumor slices?
The red and the green overlapped perfectly. They colocalized.
Wow.
Yeah, because the red dye was glowing brilliantly from the exact same physical coordinates as the green dye. It proved undeniably that this excess SQL isn't floating around in the bloodstream or hiding in the connective tissue. It is being massively produced inside the cancer cells themselves. The tumor is manufacturing its own endless supply.
Okay, so it's undeniably at the scene. But you still haven't proven it's committing the crime.
True.
How do you prove that this specific enzyme is what makes the tumor multiply?
By turning it off and watching what happens.
Ah, the classic off switch.
Right. They took living, growing human liver cancer cells in a Petri dish. To ensure their findings were universal and not just a fluke, they used two distinct types of liver cancer cell lines. One called LIHC, which is a hepatoblastoma line, and another called SKHEP1.
Okay, so two different cell lines to be thorough.
Yes. Then they used a genetic engineering technique called shRNA knockdown. In plain English, they delivered a genetic payload that specifically silenced the instructions for making SQLE. They effectively fired the factory manager.
Okay, so what happened to the factory?
The assembly line ground to a halt. They used something called a CCK8 assay to measure cell proliferation, and the growth rate of the cancer cells just plummeted.
Really? Just stopped?
Basically, yeah. Without SQLE, the cells struggled to divide. They struggled to clump together to form new tumor colonies. But the most profound, undeniable physical proof came from something called a wound healing assay.
Oh, this is fascinating. Walk us through exactly how a wound healing assay works, because it paints such a vivid picture of how cancer actually kills.
The ultimate danger of cancer isn't just that it grows, it's that it spreads.
Yeah.
It metastasizes. And to spread, a cancer cell literally has to be able to crawl through your tissue.
Right.
In a wound healing assay, scientists take a dense layer of cancer cells in a dish and literally scratch a line down the middle with a microscopic tool. They create a wound or an empty gap.
And then you just set a timer and watch.
Yes. Normal, highly aggressive liver cancer cells will rapidly migrate laterally. They will crawl across that empty plastic and close the gap in a matter of hours.
Because they're trying to spread.
Exactly. But the liver cancer cells where the SQLE gene had been silenced, they just sat there. They lost their ability to move.
Wow.
You are physically watching under a microscope as a cancer cell is stripped of its ability to migrate and spread entirely because one single metabolic enzyme was turned off.
That is just, it's incredible.
Okay,
let's take a breath and synthesize what we've learned in this second part.
Sure.
We started with computer data that flagged this cholesterol enzyme as suspicious. Then we moved to the physical proof. Bullet 1. We saw actual human tissue glowing red, proving the tumor manufactures this enzyme internally.
Perfect overlap.
Bullet 2. We used the CCK8 assay and watched living cells lose their ability to multiply when we knocked the gene down. And bullet 3, that crazy wound healing assay where we watched the cancer cells literally paralyzed in a petri dish, unable to crawl.
Exactly.
So for anyone listening who loves medical innovation, knowing that turning off this enzyme stops the cells from moving, can you imagine the therapeutic potential of a drug that mimics this exact knockdown?
It's massive.
It is. We're looking at a massive flashing neon sign pointing toward a new therapeutic target. But to invent a drug that mimics this, we need to know what the enzyme is actually doing inside the cell. Like, what is the manager yelling at the workers on the floor?
To figure that out, the researchers mapped out the protein and gene interaction networks, the PPI and GGI networks. They looked at what other molecules SQLE talks to.
Who are its friends, basically?
Right. Unsurprisingly, it acts as the master conductor for a massive cholesterol assembly line. It constantly communicates with a network of genes, things like FDFT1, LSS, CYP51A1, and a key transcription factor called SREBF2 to hyperdrive the production of cholesterol.
And we know tumors need massive amounts of cholesterol because they're multiplying so fast, right?
Yeah.
And every new cell needs a flexible, cholesterol-rich cell membrane to survive. It's basically building materials.
Exactly. It's a phenomenon called metabolic reprogramming. The tumor hijacks the body's normal systems to feed its own greed. But that is only part of the story.
There's more.
Oh, yeah. The researchers ran an analysis called Gene Set Enrichment Analysis, or GSEA, to see what broader biological pathways were being triggered. And they found something that completely shocked them. SQLE wasn't just managing metabolism, it was manipulating epigenetics.
The epigenetics.
Specifically, a mechanism called M6A RNA modification.
Okay, epigenetics and RNA. We need a solid analogy here to make sure we don't lose the forest for the trees. Let's think of a cell like a massive construction site.
Okay, I'm with you.
Your DNA is the master rule book locked in the architect's office. It never leaves. Your RNA is the photocopied blueprint that a runner carries out to the actual construction workers on the site so they know what proteins to build.
That's a great way to put it.
And epigenetics, like this M6A modification, are essentially molecular sticky notes slapped onto those blueprints. The original master rulebook in the office doesn't change, but these sticky notes tell the construction workers, hey, read this section of the blueprint twice, or ignore this page entirely, or shred this blueprint right now.
That is a phenomenal way to visualize it. M6A is the most common way our bodies naturally use those sticky notes to finely tune how genes are expressed without actually altering our underlying DNA.
Right.
But what the researchers found was a massive positive correlation between the SQLE enzyme and a very specific gene called VERMA.
VERMA. And what does VERMA do?
VERMA is the writer. It is the enzyme whose entire job is to slap those sticky notes onto the RNA blueprints. The data showed that high levels of our rogue manager, SQLE, are deeply connected to high levels of VERMA.
Oh, no.
And just like SQLE, high VERMA expression leads to terrible patient survival rates.
So the cholesterol factory manager is essentially colluding with this epigenetic writer to slap corrupt sticky notes onto the blueprints, altering the entire behavior of the cell.
Yeah.
That is terrifyingly clever.
It is deeply synergistic. And the GSEA analysis revealed one more devastating pathway being activated, the DNA damage response, or DDR.
Okay, DNA damage. You mentioned earlier that tumors with high SQLE showed heavily activated DNA repair and damage signaling. But why is the DNA getting damaged in the first place? How does making too much cholesterol literally break the DNA inside the architect's office?
It comes down to stress. Imagine a factory operating at 300% capacity. The machines are going to overheat.
Yeah.
When SQLE hyperdrives lipid and cholesterol metabolism, it creates a massive biological traffic jam inside the cell. This excessive lipid accumulation creates severe oxidative stress.
Meaning you get reactive oxygen species, or ROS, these highly unstable molecules bouncing around inside the cell like pinballs.
Exactly. And those metabolic pinballs physically slam into the DNA, snapping the strands. The activation of the DNA damage response pathway tells us that these cancer cells are constantly experiencing structural damage.
But wait, wouldn't that kill the cell?
You'd think so. But here is the twisted part. This genomic instability actually helps the tumor. Constant, low-level DNA damage forces the cell to mutate. It evolves, it becomes more resilient, and tragically more resistant to chemotherapy.
Wow. Okay, let's do our third quick mental check-in here.
Let's do it.
We are looking at a single enzyme orchestrating three massive avenues of destruction. Bullet 1. It talks to genes like SREBF2 to hyperdrive cholesterol production to physically build new tumor cells. Bullet 2. It colludes with the VERMA writer to stick corrupt M6A epigenetic notes onto the cellular blueprints. And bullet 3, the metabolic stress it creates acts like a pinball machine, snapping DNA and forcing the tumor to mutate and evolve faster.
That's the triad of destruction right there.
If you're listening, think about how wild that is. Which of these three cellular hijacks surprises you the most in a liver cancer model? Because to me, a cholesterol enzyme rewriting genetic blueprints and forcing evolution is just mind-blowing.
It really is a paradigm shift in how we view metabolism.
But, this raises a glaring, massive question for me. If this cancer cell is so wildly mutated, if its metabolism is running at 300% and its DNA is snapping, why doesn't the body's immune system notice all this chaos?
The million-dollar question.
Right. I mean, the immune system is the police force. If a factory is emitting toxic smoke and mutating wildly, the immune cops should kick the door down and destroy the place.
They absolutely should. But this brings us to perhaps the most insidious discovery in the entire study. SQLE actually functions as an immune cloak.
An immune cloak.
Yes. To understand this, researchers analyzed the tumor immune microenvironment, or the TIME. They used an algorithm to map out exactly which immune cells were infiltrating these liver tumors.
And what happens when a tumor is flooded with SQLE?
The entire immune landscape flips upside down. Let's talk about T cells. In a healthy response to cancer, your body deploys Th1 cells. These are your attackers. They coordinate the assault on the tumor.
You'll get them.
But the data showed that high SQLE causes a massive spike in Th2 cells instead. Th2 cells are immunosuppressive. They secrete biochemical signals, like cytokines, that actually protect the tumor and calm down the attackers.
Oh, wow. So the tumor is essentially hiring corrupt security guards to tell the real cops everything is fine.
Yes, and it gets worse. It doesn't just hire suppressors, it actively starves out the heavy hitters. The researchers looked at CD8 plus T cells.
Those are the big ones, right?
Yeah, these are the immune system's primary assassins. The cytotoxic killers meant to hunt down cancer. Tumors with high SQLE showed a widespread, massive drop in the infiltration of CD8 plus T cells, as well as natural killer cells and B cells.
And this wasn't just a computer projection, right? They went back to those physical human tissue samples.
They did. They used the fluorescent tags again. This time, they stained the tumor cells hoarding SQLE in red, and they stained the CD8 plus T cell assassins in yellow.
Okay, red tumor, yellow assassins. What did it look like?
The visual evidence was incredibly stark. When you looked at a tumor with high SQLE, just a glowing sea of red, there were almost zero yellow CD8 plus T cells to be found. They had an inverse spatial relationship.
But how? How does a cholesterol enzyme physically stop an immune assassin from entering the tumor? Like, what's the actual mechanism?
It goes back to that metabolic traffic jam. By holding lipids and rapidly altering the microenvironment around the tumor, SQLE creates a localized zone of metabolic exhaustion.
Okay, what does that mean for the T cell?
Well, the CD8 plus T cells need energy to fight. But the tumor has fundamentally altered the chemical environment. It's acidic and depleted of normal nutrients. When the T cell is trying to enter this lipid-dense acidic barrier, they literally run out of fuel. They become exhausted and fail to penetrate. SQLE basically builds an invisible, immune cold force field.
That is it's a huge light bulb moment.
Isn't it?
Because in the news, we constantly hear about immunotherapy as being the holy grail of cancer treatment. We are literally engineering T cells in labs to attack specific tumors. But this explains exactly why those miracle therapies so often fail in advanced liver cancer.
Precisely.
You can engineer the greatest T cell assassin in human history, but if the tumor has used a cholesterol enzyme to build a metabolic brick wall, the T cells just bounce off. They starve before they can take the shot.
That's the reality. This study proves that you cannot divorce metabolism from immunology. The metabolic reprogramming driven by SQLE directly dictates the immunological failure of the patient.
Okay, let's do our final mini recap here. We've got our immune cloak. Bullet 1, SQLE spikes Th2 cells, the corrupt security guards. Bullet 2, it starves out the CD8 plus assassins. And bullet 3, we saw visual proof of this inverse relationship where the yellow T cells just couldn't penetrate the red tumor.
Perfectly summarized.
So listener, ask yourself, if a tumor can use a cholesterol enzyme to build an invisible shield, how does that change how we think about the relationship between diet, metabolism, and immunity? It's all connected.
It really is. If we connect this to the bigger picture, SQLE is not just a biomarker. It is a metabolic driver, an epigenetic manipulator, and an immune suppressor in LIHC. The depth of this data fundamentally redefines our understanding of liver cancer.
It's unbelievable.
And it provides a highly robust foundation for developing personalized treatments that could dismantle this tumor from the inside out.
Well, let's pull all of this together. We have journeyed through an incredible piece of scientific detective work today. We started with massive mathematical sieves catching one suspicious gene out of thousands. We saw actual human tissue glowing red, proving the tumor manufactures this enzyme.
Yep.
We watched cancer cells paralyzed in a petri dish when we turned the gene off. And finally, we learned how this single enzyme, squalene epoxidase, acts as a master villain. Thank you so much for guiding us through the intricate details of this machinery.
It's been my pleasure.
And a huge thank you to you, our listener, for joining us on this deep dive. We know how complex these biological pathways are, and we love taking the time to share these deep aha moments with you.
Absolutely.
But before we wrap up, I want to leave you with one final lingering thought to mull over. We established today that this highly aggressive, deadly liver cancer hides behind an impenetrable shield. And that shield is built by a totally standard rate-limiting enzyme in your cholesterol biosynthesis pathway. So, think about this. Could existing, incredibly cheap, widely available cholesterol-lowering drugs like the statins millions of people take every day be repurposed? Could we theoretically use a simple cholesterol pill to inhibit SQLE, strip away that metabolic armor, and make these cold, impenetrable tumors hot again for immunotherapy?
It's a fascinating question.
Keep that in mind the next time you hear a conversation about metabolic health.
Yeah.
There is always more beneath the surface.